The next speaker is Dr. Sharma himself. Dr. Praveen Sharma has more than 35 years of experience in teaching clinical biochemistry and laboratory management. He has more than 80 research publications in various prestigious journals and has interest in diabetes, metabolic syndrome, atherosclerosis, antioxidants, lead toxicity. He has delivered invited lectures and chaired sessions in different national and international meetings. He is a recipient of GS Seth Medical College, Pattabhiraman and Avat Saran Memorial Oration Awards. He has been awarded with the Merit Award of Rajasthan Government in 2002. He is the past president and fellow of the Association of Clinical Biochemists of India. He also served as the member of the Accreditation Committee of National Accreditation Board for testing and calibration of laboratories during 2004-05 and is an NABL assessor. He is a ACBA representative to the Asia Pacific Federation of Clinical Biochemistry and Laboratory Medicine, Education Committee and National Representative of IFTC. He is Editor-in-Chief, Indian Journal of Clinical Biochemistry and Chairperson, Corporate Wing of ACBI. Dr. Sharma is the Chief Communication Committee of APFCB and is also Chief Editor of APFCB News. Dr. Sharma is the Director of National Referral Center for RED Projects in India, Jodhpur. Presently, he is Professor and Head of the Department of Biochemistry, All India Institute of Medical Sciences, Jodhpur, and President of Association of Clinical Biochemists of India. Dr. Sharma.
So we have been talking about what the reason for obesity in this country. What are the important and what are the important determinants for obesity in this country? What is the role of overnutrition? What is the role of undernutrition? And what is the nutrition in this country? Naturally, when we talk about the obesity, we need to think about where obesity. It is abdominal obesity, the whole body obesity. What we are talking about? Visceral obesity, the obesity. So something has to do with adipocytes. So I will be talking about adipocytes and what are the changes, what are the important compounds dependent from adipocytes which could create insulin resistance, one of the important cause of diabetes and atherosclerosis, cardiovascular disease.
देखिए सर इसको करो ना प्रयास करता हूँ इसको करो ना यार हो जाएगा अब ऑफिस में सेट करना होगा तो कर
So adipocytes are red cells, approximately 1/3 of are mature adipocytes and rest all 2/3 are various premature cells, includes small parenchymal stem cells, regulatory cells, endothelial cells, macrophages and pre-adipocytes. Adipose tissue is characterized by marked cellular heterogeneity Adipocytes, as I said, they include adipocyte, fibrocyte, and different type of premature cells. Many transcriptional factors are required to convert premature adipocyte to mature adipocytes. The development of fully differentiated mature adipocytes from pre-adipocytes involve the sequential activation of using several transcriptional factors. As we all know, adipocytes, they are present throughout the body in loose connective tissues. They are of two types, white adipocytes and brown adipocytes. Brown adipocytes are essentially involved in thermogenic processes, whereas white adipocytes are used for the energy storage. Obesity can be, if you talk about the obesity, what happens in adipose tissue during in obesity? We need to know this, a few of the important facts. Obesity can be, through hyperplasia or hypertrophy. What is the fate of lipid metabolism? In insulin resistant cells, we all know that more of the fatty acid will be produced and fatty acid is spilled in circulation, leading to lipotoxicity. There is unregulated production of adipokines. There is an infiltration of adipose tissue by macrophages. Then there is a decreased adipose tissue blood flow, and this may lead to ER dysfunction and hypoxia-like conditions. So this is the adipocyte composition. If you look at this is the adipocyte composition, and then adipocyte, there is hypertrophic growth through hypertrophy or hyperplasia. Hyperplasia means there is an increase in the number of adipocytes, whereas hypertrophy, it's an increase in size of adipocyte. In most of visceral tissues, adipocytes grow by hypertrophy, whereas in subcutaneous tissue, their growth is through hyperplasia. So this clearly shows that adipocyte hyperplasia is predominant in subcutaneous as compared to in visceral tissues, particularly in obesity. And hypertrophic growth is more in visceral tissues, and which is directly proportional to the BMI. Adipose tissue is the major fat storage unit in the body. 90% of lipids are stored in the form of triglycerides. If lipogenesis is less than lipolysis, then the free fatty acids are released into the circulation. Chronic release of free fatty acid to produce lipotoxicity in the muscle, liver, and many other tissues. Insulin resistance is one of the important conditions which may be the outcome of increased free fatty acid in the blood and liver. Intra-abdominal adiposity, which essentially visceral adiposity, causes dyslipidemia, abdominal obesity, glucose tolerance, insulin resistance, and ultimately these are the important risk factor for the development of cardiac disease. Intra-abdominal obesity can causes these is risk factor for various diseases. When there is increased production of free fatty acid, or there is increased accumulation of free fatty acid, apart from its deposition in adipose tissue, it gets deposited around the tissues and organ in various tissues, and it can cause lipotoxicity. So This is one of the examples how it causes lipotoxicity in heart, and this what happens to heart. When it deposits around the heart and heart muscle, it causes reduces cardiac contractibility and recovery from the myocardial infarction. And the mechanism include accumulation of lipid-derived intermediates, mitochondrial dysfunction, ER, stress, increased transcription, activation, inflammatory changes, not many things. And this all results into cell death and apoptosis. So adipose tissue is not merely fat storage tissue. It can produce many other things. It acts as endocrine organ that secretes many biologically active substances such as leptin, free fatty acid, tumor necrotic factors, which are collectively termed as adipokines. And these adipokines can affect different organ systems. It can cause cardiovascular disease, hypertension, reproduction, it affects immune system, it also affects lipid metabolism, glucose metabolism, and energy balance in the body. So this is normal function of adipocyte, and adipocyte generating various adipokines affecting various organs including liver, pancreas, and vasculature, muscles, etc. So various adverse cardiometabolic effects of product of adipocytes include inflammation, hypertension, or lot many things. These are various. This is further and bioactive. These bioactive substances secreted by adipocyte modulate insulin resistance and cardiovascular disease. These are again adipocyte-derived bioactive substances and their activity. Now, as I talked, what happens when cell undergoes growth through hypertrophy? It grows, it becomes big, which can rupture also. And when it ruptures, it will produce many different types of cytokines and adipokines. In obesity, what happens? Pre-adipocytes are converted into macrophages. And profile of these macrophages are, essentially similar to the pre-adipocytes. And under the right condition, they are converted into macrophages, and then macrophages infiltrate into adipocytes. Adipose tissue macrophages are present in two forms, two subsets, resident M2 type and inflammatory M1 type. M2 macrophages are responsive to IL-4 and IL-13. They secrete anti-inflammatory cytokines IL-10. Upregulated production of arginine-1 enzyme results in nitric oxide synthesis and inflammation also. M1 macrophages, as you know, they are pro-inflammatory. So as I said, macrophages originate from monocytes in the blood. They cause angiogenesis. They are also responsible for clearing of necrotic tissues. They secrete pro-inflammatory cytokines, TNF-alpha. Macrophages develop into specialized cell type with a specific function response to different stimuli. So these macrophages perform dual role, good as well as bad. As long as angiogenesis and clearing of necrotic factors are concerned, it's a beneficial role of macrophages where the production of pro-cytokines and pro-inflammatory cytokines. This is evil role of macrophages. And then this is what happens in obesity. They can be classified into two: alternatively activated macrophages and classically activated macrophages. And there is a change from alternatively activated macrophages to classically activated macrophages in obesity. There is more of this classically activated macrophages are present. which are responsible for, which promotes pro-inflammatory cytokines, and whereas alternatively activated macrophages promote angiogenesis. How does infiltration occur? Macrophages, monocytes that circulate in the vasculature. And when they leave vasculature and infiltrate into tissue, they are known as macrophages. How they enter into the cell, there is one protein, MCP. This MCP protein is responsible for this which is monocyte chemoattractant protein 1. This is responsible for infiltration of monocyte into adipocytes. It is a pro-inflammatory cytokine produced by adipose tissue, recruit monocytes, leukocytes and other inflammatory cells. It is upregulated in obesity and type 2 diabetes. So there is a direct link between MCP1 and obesity, and it is a very good marker for macrophage infiltration. So this shows that MCP1 RNA level is more level in subcutaneous adipocytes, and MCP1 plasma level in positively correlated with BMI. So infiltration rate of monocyte into the vascular adipose tissue is higher than into subcutaneous adipose tissue. MCP1 protein level is higher in visceral as compared to subcutaneous adipose tissues. And this has direct association with body weight. This is a detailed diagram how monocyte enters into adipocytes in the presence of MCP1, and there is secretion of various adipokines which are responsible for the development of insulin resistance. These cytokines that are inflammatory markers are TNF alpha, and GEN kinase controls the stress sensing pathway, stress signaling pathway. So there is a paracrine loop between the adipocytes and macrophages that aggravates inflammation changes in obese adipose tissues. A co-culture of adipocytes and macrophages results in increase in pro-inflammatory cytokines, decrease in adiponectin, and increase in free fatty acid levels. Key player of paracrine loop is TNF alpha from the macrophages and free fatty acid from adipocytes. If we compare both subcutaneous adipocytes and visceral adipocytes, we can see that fat amount is relative to total body fat. Fat is more in subcutaneous. There is a greater differentiation of pre-adipocytes in subcutaneous as compared to visceral. Apoptosis is more in visceral adipocyte as compared to subcutaneous. Leptin level is more in subcutaneous as compared to visceral. Adiponectin is equal almost. IL-6 is more, pro-inflammatory marker is more in visceral as compared to subcutaneous. And TNF alpha again is more in visceral as compared to subcutaneous adipocytes. So adipocyte dysfunction increases the circulatory concentration of free fatty acid or NAFA, which is, and in this condition, there is inability of adipose tissue to buffer the excess nutrient intake and the dyslipidemic state. That is, this state is something like metabolic syndrome. When there is overload or increased production of FOP containing particles in liver that carry triglyceride to the adipose tissue, resulting in LDL formation. This occurs in visceral adipose tissue very efficiently, and this depot is also more capable of releasing lipid in time of requirement. The subcutaneous adipose tissue has actually a more large capacity to store the lipid in given its usual large size. So this is this shows how this adipocytes secrete free fatty acid and adipokines, which can affect insulin receptor and produce insulin resistance, and this also causes apoptosis. This indicates what is the lipid metabolism, how it is affected in obesity. There is an increased production of free fatty acid in insulin resistant adipocytes, and then there is increased production of LDL, small dense LDL, which is more atherogenic, and there is a decrease in SDL, which is an increased production of cholesterol-rich VLDL, which is more atherogenic as compared to triglyceride-rich VLDL. This is how it affects different organ, increase in adipocyte or intra-abdominal obesity. So, excess free fatty acid from intra-abdominal obesity can therefore induce both insulin resistance and impaired beta cell function, the primary metabolic defect that underlies type 2 diabetes. Obesity is also associated with deregulated lipid and carbohydrate metabolism. An increase in either of these substrates increase the demand of mitochondria and utilization of electron transport chain. This creates relatively hypoxia together with increased need for nutrient oxidation. This generates unusual amount of reactive oxygen species, which is again one of the important cause for the generation of insulin resistance. So in such condition, insulin adipose tissue blood flows tremendously decrease, and adipose tissue as adipose tissue expand, vasculature may be unable to meet the oxygen demand. Adipocytes become hypoxic, leading to inflammation and cell death. This is proved by the evidence that hypoxia inducible factor HIF1, a marker for hypoxia, is correlated with decreased adiponectin and PPAR gamma, and increased PAI1 and other inflammatory markers and cytokines. And also there is a decreased blood flow, means decreased ability to clear triglycerides and waste product from the tissues. Adipose satisfaction also causes organelle dysfunction, that is ER stress and mitochondrial stress, and this cell responds by initiating an unfolding protein response where protein synthesis is inhibited, leading to clearing of abnormally folded proteins. If ER stress continues, unfolding protein response becomes overwhelmed and resulting in oxidative stress, which is important reason for the development of oxidative stress. If UPR is not relieved, ER stress will lead to cell death and apoptosis. And then portal theory against, there is increased production of free fatty acid leading to insulin resistance. Visceral adiposity predicts insulin, impaired glucose tolerance, hypertension, dyslipidemia. Visceral fat is highly active metabolically, more susceptible to lipolysis than subcutaneous fat, but it is weak producer of adiponectin. There is a positive correlation between adipocyte size and TNF-alpha, IL-6 and HSCRP, while negative correlation with adiponectin. Why do we are concerned about visceral adipose tissues? So there is imbalance in this tissue in metabolic syndrome. Visceral adipose tissues play a good or bad role. It makes beneficial compound which raise sensitivity to insulin and reduce blood pressure and sustain healthy blood vessels. But it also makes compounds that promote inflammation. When the fat is healthy, it predominantly makes the beneficial compound, and when it is unhealthy, there is a shift from inflammatory compounds, shift to inflammatory compounds. So visceral adiposity seems to play an important central role in metabolic syndrome, being generally considered much more pro-inflammatory conditions. As compared to the fat accumulation in abdominal cavity, the size of the abdominal adipocyte is more important. Larger adipocytes are more prone to the rupture, and cell rupture constitute a focus on inflammation. Macrophages crowns surrounding by dead adipocyte, crowns surrounding dead adipocyte in adipose tissue are compatible with this hypothesis. Now, what happens to adipocytes when it grows? In obese, in lean and normal, a person with a lean weight and normal metabolic function, there is no inflammation. Metabolically it is controlled, there is vasculature is normal. Where with gradual increase in obesity, it becomes more there is more inflammation, decreased metabolic control, there is decrease in vascular function, and anti-inflammatory markers which were predominantly present in normal condition have now been changed to pro-inflammatory markers. And macrophages, M2 macrophages, there is a change into M1 macrophages, helper cells have been converted into killer cells. So another characteristic of abdominal adipose tissue is its high metabolic activity, dense vascularization. On the other hand, increased intra-abdominal pressure as well as pressure variations may easily create period or zone of hypoxia. Vascular endothelial growth factors, leptin, adenosine, substance P are important substances produces in this feature. Further, there is hypoxia by itself inactivating the adiponectin promoters. This is overall metabolism that occurs in obesity, producing cytokines, adipokines. So, there is a chronic inflammation leading to production of various pro-inflammatory markers. There is insulin resistance, dyslipidemia, there may be cancer in obesity, infertility, many things associated with obesity. But obesity is also associated with all of this, so it is very difficult to say whether it is inflammation is the cause or it is obesity is the cause. Thank you very much. क्या करके लाया साले को पकड़
The next speaker in this session is Dr. Kuldeep Singh. Dr. Kuldeep Singh did his MBBS and MD in pediatrics from KGMC Lucknow in 1991. He completed his DM in genetics from SGPGI Lucknow in 1995. He was awarded the fellowship in Academy of Medical Sciences 2014. He has a special interest in medical education with many publications in nutrition and international journals up to his credit. He received the traveling fellowship to International Societies of Human Genetics, Austria in 2001. He also received the MAMS award in 2014. Presently, he is additional professor in pediatrics at AIIMS, Jodhpur. Dr. Kuldeep Singh.
Thank you, chairpersons, and all the delegates who are attending this symposium, and dear students. We have now this understanding the causes of obesity. So in the sequence of these causes, one of the important aspect is what are the role of the genes, whether the genes are there or it is an environment. There are long research which is still going on. So today I will be discussing about this molecular genetics of the adiposiness, adiposity, and some of the aspect of the metabolic syndrome. The objective of my talk will be to emphasize what is the role of the genetics which plays a major role in adiposiness, adiposity, and metabolic syndrome, to know how the genes affect the obesity, and identify some of the mechanism by which gene influence a person's risk of becoming obese. So there is a genetic basis of the obesity, and now we know that there is an epidemic going on, which is of the obesity. The people are becoming more obese. The Western world has already engulfed by that obesity epidemic. Now it is engulfing our country also. There are many discovery of the genes which are responsible for the obesity. There are many association studies of the genes which indicate that there is a genes for obesity. Some of the disorders may follow the monogenic genetic disorder, and there is a genetics of the metabolic syndrome. So much so that this issue was totally dedicated to the obese species. Obesity is defined as body mass index of more than 95th percentile for the age and sex of that individual. And there is a National Health Nutrition Examination Survey in US which said that there was a 16% overweight and that 31% of the population are at risk. Since the previous survey, there is a more than 300% increase in the obesity and overweight. But usually these surveys are mainly dominated by the environmental drivers. They were looking into the environmental factor which are responsible for the obesity. Now this, you can see that there is a fatty tissue and there is a thinness which constitute the constitution of an individual, which can get aggravated because of the genes environment interaction. And now these genes environment interaction leads to increase in the weight, and ultimately the patient becomes so obese that the intervention has to be done. At that intervention point, there is a adipose tissue had markedly changed. If there is angiogenesis in this adipose tissue, it is protective, but if there is no angiogenesis, it is not protective and it becomes more of the macrophage and there is more of the inflammatory response. When we try to do the intervention, there is some reduction, but it is not sustained properly and it again revert back to the original status. So the genes are important, which can result into the continuing obesity despite the environmental reduction. And there are some epigenetic events. Epigenetic events are those genes changes which are occurring in some other genes but can affect the genes in their protein synthesis or their protein formation and functioning. These are known as epigenetic phenomena. So there are now many epigenetic phenomena also responsible for the gene interaction. Till now we know that there is a individual psychology, food consumption, environmental, social influences and the activity which determine the obesity. But now we know that major part of this is determined by the biology of the individual. The role of the genetics is very important because it is more powerful for those who have a severe and early obesity. And identification of this critical molecular component of the human control systems will help in the development of the specific drug which can decrease the obesity. The genetic is also having a role in the makeup of the individual to influence his or her response to the treatment measures, and it also helps in identifying subgroups of people who might be responsive or resistant to the environmental modulations. On one extreme there is a gene, on the other extreme there is environment, and here there are monogenic which are highly predicted gene. On the other hand, there is multiple genes are involved in the obesity. Now recently term has been coined as omics, proteomics, omics. Now omics is the total particles which are there in the metabolism, which are of very small quantities, but with the huge bioinformatics system, the scientists have been able to assemble all these into single thing which can be predictive of the drug or the occurrence or the prediction of the obesity. Twin studies have indicated there is a 70% heritability if the genes are there in the twins, and the concordant is what the conjoined monozygotic twin is 0.7 to 0.9%. So if we see the obesity as a whole, it can be non-syndromic or syndromic. In the non-syndromic, it can be monogenic obesity or it can be polygenic obesity. In the monogenic obesity, these are the markers or the single nucleotide polymorphism or the genes LAP, LFR, POMC, MC4R, PC1 and PY and S1, M1. In the polygenic, similarly, they are dependent on the adrenoceptors and these 1, 2 and 3, and the uncoupling proteins of the mitochondrial system 1, 2 or 3. Whereas the syndromic variety can be of the pleiotropic syndromes. They are although rare, but they have been extensively studied. And nowadays we are coming across some of the chromosomal rearrangement. So on one hand there is a rare monogenic mutation in which the single gene is detected, and we can find out what is the mutation there. On the other hand, in the polygenic there is a challenge because there is a pertinent genes may be there, and there is a lot of experiments have been done in the last 25 years. Many new mutations and genes have been identified, and they are all dependent, they are interdependent. You can see they are all interdependent, and they are also dependent on the environment. On one hand there is increased energy, and on the other hand decrease energy or the expenditure. So ultimately they react together, but one good thing is that we cannot say that the information which was collected earlier is waste. Now this, with the help of bioinformatics, can be more utilized in a more better way to predict the obesity. Another important part of the genes is that the body weight regulation depends on the food intake, energy expenditure and the adipogenesis. There are some genes which regulate the food intake. Now these are the genes encoding the leptin. There is a leptin and ghrelin, which are the enzymes which are acting on the brain to increase the satiety or increase the hunger or suppress the hunger. And there is a mutation in the pro-opiomelanocortin, POMC. This gene mutation has shown that if it occurs in the newborn, it may lead to the ACTH deficiency, which is characterized by red hair and the pale skin. Then the mutations in the melanocortin 4 receptor, MC4R. This is very important because it is in between the monogenic as well as polygenic. They have a hyperphagia, accelerated linear growth and increased bone density. This picture shows that in the brain there is an arcuate nucleus and the paraventricular nucleus. So here the leptin genes are related and POMC genes are related to the arcuate nucleus, whereas the MSG gene, MC4R gene and SM1 gene is related to the paraventricular nucleus, and they depend, the phenotypes depends on the, if this is active or it is deleted, then there is a obese phenotypes result. And these are the conditions which relate to rare human phenotype, but here this is a polygenic disorders and obesity only may be the manifestation. Only obesity may be the manifestation. Another groups of genes are which regulate the food intake, and it has been found in the UK study that 50% of the populations are more likely to be obese because they have a particular mutation in MC4R mutation. Now this variation has also been commonly found in Indian Asian ancestry. So that's why this may explain that why the Indians are also predisposed to obesity and may develop obesity if the environment is conducive. These gene variants also make people more likely to develop insulin resistance and the type 2 diabetes, that is, they can go into the metabolic syndrome. There are some genes which regulate the food intake like PC1, NPY, ghrelin receptor gene, and about 40 to 80 genes are there which are protein coupled receptors in the taste cells and which may alter our preference to the food. So one may prefer more food and may lead to obesity. One of the most important gene which is being very commonly studied is FTO, that is fat mass and obesity associated genes. Now this regulates the ghrelin hormone, which is an appetite hormone responsible for eating behavior. Ghrelin stimulates the hunger and increases the food intake. It also reduces the sense of fullness after eating, causing further eating, and it also causes a preference for high fat food. On the other hand, there is a high risk variant of the FTO gene which actually works in an epigenetic way. That is, it is not directly involving the genes which are leading to obesity, but epigenetic way by removing the methyl groups. Now this is a reason for occurrence of the genomic imprinting, that the genes can imprinted because of the epigenetic phenomena, and they, even if there is no mutation, because of the methylation, they may abnormally behave and lead to ghrelin production. There are some genes which are encoding for energy expenditure because we know that there is a thermogenesis which is adaptive in nature in the humans, not in other animals, and that is mediated by the sympathetic system. So the sympathetic system are regulated by these genes, ADRB2, ADRB3 and B1. They are the candidate gene for the obesity. Similarly, uncoupling proteins which are encoded by UCP genes in the mitochondria are important for the energy efficiency. Other genes which are important in the implicated in the adipogenesis are transcription factor for the adipocyte differentiation, and there is a key factor which is known as peroxisome proliferator activated receptor gamma or G, PPARG gene, which is important for the adipogenesis, and this gene variants may also be functionally reduced in the activity. So even the functional reduction may lead to the obesity. Now what about the syndromic obesity? We are usually listening about the syndromes, and the Bardet-Biedl syndrome is the heavily studied syndrome for the obesity. This Bardet-Biedl syndrome may be associated with the polydactyly in the Lawrence-Boone-Biedl-Bardet syndrome. There is Albright hereditary osteodystrophy is also associated with ectopic calcification ossification. Borjeson, Forsman and Lehmann syndrome is associated with the mental retardation, gynecomastia and hypogonadism. Cohen syndrome is also a result of the obesity, microcephaly, and there is a prominent upper incisor. This is a table which shows the feature of the Prader-Willi syndrome. Prader-Willi syndrome is one of the microdeletion syndrome which is characterized by muscular hypotonia, low birth weight, but the weight increases by the end of the 1 year, and they become so obese that they are the candidate for the surgery. Bardet syndrome, Elstrom syndrome and Borjeson Forsman Lehmann syndrome, and these are the loci which have been mapped. Now other monogenic obesities are because of fragile syndrome also. Even in the chromosomal disorder like Down syndrome, they are predisposed to obesity. Ulnar memory syndrome is characterized by defect in the chromosome 12, and there is ulnar hypoplasia with the gynecological abnormality. Simpson-Gulabi-Behmel syndrome is also X-linked disorder which is of overgrowth disorder and associated with the visceral abnormalities. MAHMO, that is mental retardation, epilepsy, hypogenitalism, microcephaly and obesity. It is an X-linked recessive disorder. I talk about the Prader-Willi syndrome. This is because of the chromosomal rearrangement and there is a genomic imprinting. The same mutation, if it is passed from father, it leads to Prader-Willi syndrome. Whereas the same mutation, if it is passed from the mother, it leads to Angelman syndrome. So how the same mutation or same deletion can have the different manifestation is because of the genomic imprinting, and there are some epigenetic phenomena. Balanced translocation of this chromosome between 1 and 6 lead to disruption of the S1M1 gene which is present on chromosome 6. So chromosome 6 is also the candidate for the genes abnormality which may lead to obesity. Apart from this monogenic disorder, there is a complex traits, and there is a many candidate genes which are related to the these obese phenotypes, and there are more than 400 studies covering 113 candidate genes which have reported in association. Now, I was talking about the association in the single gene. When the disease is being identified in a single family and we identify in other individual with the help of linkage analysis, this is known as family-based linkage analysis. Whereas if the disease is studied in the population and some of the person may be carrying that gene, but the majority of the with the disease will be carrying the gene. So that means there is association between the gene and that trait. Whereas here it is not an association, it is by chance. But because there may be some other factor which are regulating the obesity, so it may be not occurring. So these are known as genome-wide association studies. Why the genome-wide association studies is when you want to investigate the entire genome to determine what polymorphisms are associated with the condition you are studying. The advantage of this is that the biology of the condition does not need to be the biology of the condition does not need to be well understood. You can investigate the entire genome and see where the data takes you. But the disadvantage is the large number of the subject is required. So nowadays, genome-wide association studies with the help of bioinformatics markers, we are coming close to the genes which are involved in the obesity. Now there are 43 genes for BMI, overweight. There is a 13 genes for body composition, fat distribution 12 genes, energy expenditure 4 genes, change in the body weight 7 genes. Negative association also exists with the genes. Now these are the biomarkers. The genes can also be labeled as a biomarker, and this availability of these biomarkers may greatly facilitate the therapeutic intervention. And in addition, they are also important for subclassifying these obesity disorder. As you can see that obesity is there is a one spectrum. There is a single genes monogenic disorder, or there is a polygenic disorder. Now these genes are also associated with some other like oncogenes, tumor suppressor genes. So obesity may lead to other problems also, comorbidities, cancer, respiratory problem, bone growth abnormalities, diabetic complications, and the insulin resistance. These are the list of the genes which have been studied as the biomarker. So there is a firm evidence that the genetic susceptibility to obesity can be blunted in part through physical activity. It is not that if the gene is there, they will always develop obesity. But with the physical activity, and there is a strong evidence that it can control the obesity. There are 3 studies which suggest that the high fat diet can amplify the effects of the FTO genotype. So if the high fat is taken by the individual who is having the FTO genotype, there is a development of the obesity. On the other hand, if he decreases the high fat diet, there will be a decrease in the obesity. Obesogenic environment may amplify MC4R pathogenic monogenic mutation. So it is the presence of the environment also which is important. Now there are some genes which influence the response, MC4R, POMC, FTO genes. So personalized medicine is only possible till now for a condition known as congenital leptin deficiency. Now this congenital leptin deficiency, if this leptin can be given in the external form, it will lead to the decrease in the obesity or normalization of the person. But this is an extremely rare condition. Until now only 14 case reports have been published. Now one of the important concept of the obesity is the metabolic syndrome. Now this is very interesting. Metabolic syndrome is determined by the interplay of the genetic and the environmental factor. And all the components of the metabolic syndromes are strongly inherited. All the 3 components of the metabolic syndromes are strongly inherited. And there is a condition known as, or there is a hypothesis known as thrifty genotype hypothesis. Thrifty means is a conjuicy, or they want to keep the things to themselves. So thrifty genotype hypothesis proposed that energy conserving genotypes selected by a harsh environment are associated with a survival disadvantage when there is an abundance of the food. So in the older time, when there was a scarcity of the food, this helped us to survive. But now when there is no scarcity, it is causing the problems. So this is a thrifty genotype hypothesis. The genes want to still preserve whatever amount of the intake is there. Another concept is the thrifty phenotype hypothesis, which suggests that the intrauterine malnutrition, this is also very important, intrauterine malnutrition leads to low birth weight and increased risk of metabolic syndrome later in the life. It has been proposed by the Barker and later confirmed by the Gluckman, and they said that if the intrauterine environment is abnormal, that is there is a IUGR, then the metabolic system of the baby continues to behave abnormally even if the correction of the abnormality postnatally. So that will lead to metabolic syndrome at the early age. It is also known as the adult onset disease in the infancy or the childhood, or it is also termed as the child is a father of man. That is the disease of the older people occur at an earlier age. So even a 1-year-old child becomes so fat if the environment correction occurs and if there is intrauterine malnutrition. So genomic scans have identified. Now genomic scan is again the genome wise association studies, and they have been pulled together, so that they are recognized as the scans, and there is a strong links between the certain chromosomal region and the metabolic syndrome. And the candidate genes have identified linkage between metabolic syndrome and a number of genes. Now these are some of the genes: PPARG, adenosine, CD36 and beta receptor. So now we can see that we are studying the genes, the picture was not clear, but gradually some of the genes can be more focused, and gradually they become more focused. The picture is still blurred, but gradually because of the more of the mutations and the bioinformatics we are applying to the system, that is the picture later on becomes clear. So we are moving from the experiment to the clearer picture, and now the picture is more clearer. That means that the experiment which were done earlier has not gone waste. They are still there, and if we can use them, we can make the picture very clear about the genes resulting into obesity. I acknowledge gratefully the National Academy of Medical Science for the funding, and Professor J.S. Bajaj for mentoring for this symposium. Thank you.
May I now request Dr. Kuldeep Singh to present a token of appreciation to Dr. Prema Ramachandran. Thank you, sir. Thank you, ma'am. We now have a break for tea, and we return back after 15 minutes for the next session.